Thank you. Yeah, I'm really happy to be here. And I also want to particularly thank Ariane and Miriam and Nikolai and Agnes for the invitation. It's been really fun. And I think my terrible French has maybe improved a teeny bit since I listened all day and tried to pick up a few words. So what I wanted to do today was tell you a few short vignettes of stories that are examples of how we as developmental and stem cell biologists can think about taking some fundamental principles and applying them towards understanding and looking at therapies for rare diseases. So our lab is generally interested in how organs form in embryos and how they renew and regenerate in the adult. And we focus a lot on the craniofacial complex as well as the gut. And so what I broke this down into was 4 short segments. The first 3 are craniofacial, and then I put in one at the end about the gut just to give you a sense of what we work on. And each of them has as a core a rare disease that we think about. And mostly they're fundamental mechanistic studies, and some of them move into therapies as well. So the first condition I want to talk about is one that I'm sure is familiar to most of you in the audience, which is ectodermal dysplasia. And this is actually a large group of diseases, many dozens of them, and they fall into several different categories. And one of the best known categories are the ones that are in the ectodysplasin pathway. And so this is the ectodysplasin ligand is a member of the TNF alpha superfamily, and then there's a receptor and intercellular signaling parts. The ligand is on the X chromosome, so that's why mutations in this gene are cause a condition called X-linked hypohidrotic ectodermal dysplasia, or XLHED. And this is a classic triad of the reduced ability to sweat, misshapen or absent teeth, and thin or sparse hair. There's actually, this was described many centuries ago now, and there's a very nice description by Darwin in one of his books of an Indian family in which there were 10 men, so this is the X-linked part, over the course of 4 generations, that's the inherited part, and they have abnormal teeth and hair, and they don't sweat correctly. So really a very nice description quite a long time ago by Darwin. And then mouse mutants were identified for various components of the pathway as spontaneous mutants in the 1950s. TABI is the one for the ectodysplasin or EDA1 gene. The gene was cloned in humans over 20 years ago. Is that right? Yeah, over 20 years ago. And then the first clinical trial for safety was actually done at UCSF for a replacement molecule, which I'll tell you about over the next couple of slides. And what I think is particularly interesting about this condition is that it's the treatment approach is really paradigm shifting, because it's the first attempt, to my knowledge, to treat a structural birth defect with a medicine instead of a surgery. If you think about any of the other diseases that we talk about in which we're trying to correct structural abnormalities like cleft lip and palate or congenital heart disease, they're approached surgically, and this is now something that people are thinking about doing with a medicine. And this is the credit for this all goes to Pascal Schneider, who's in Switzerland, who came up with the idea of taking a tabby mouse, the ectodysplasin mouse, carrying an affected embryo. And for those of you who haven't studied this condition before, this is what the untreated animal looks like. So it has abnormal fur and skin and teeth. And then Pascal made a recombinant version of the ectodysplasin protein and administered it to these pregnant females, and is transplacentally transmitted. And what that led to was essentially a complete recovery, so normal hair and skin and teeth. And he published this now 20 years ago. And then over the course of a number of years, there was an attempt to move this first into a dog model. And in the dog model, because dogs have primary and secondary dentition, unlike mice, which just have primary dentition, it was actually given postnatally. And so then the idea was, could you also treat humans postnatally? And so we at UCSF as part of a large group, including the Necker Hospital here, began to do safety studies, and it turned out to be safe in humans. And then we actually dosed the first U.S. baby postnatally, and we did a large number of studies, including looking at sweat glands and assessing the hair. And to boil down a very long series of studies, unfortunately, it looked like giving the drug postnatally to humans did not work. So this was a big bummer, and the company that was doing this ran through close to $50 million and actually ended up going bankrupt. And so it was a very sad end to this story, we thought, because the company was gone and we couldn't continue this. But fortunately, this guy named Holmschneider in Germany, not related to Pascal Schneider, but a close collaborator of his, who has really focused his entire life on studying ectodermal dysplasia, knew that there was a little bit of the drug still left, even though the company had gone bankrupt. And he said, well, maybe the problem here was that we're giving it postnatally instead of prenatally. And so he then identified some pregnant carriers of this, and this is now published a few years ago, showed that by giving the drug prenatally, he could actually correct many aspects of the disease. So this was a very exciting development, and like I said, really I think the first attempt to treat one of these structural birth defects. And we can talk about why ectodermal dysplasia is such a great candidate, unlike some of the other structural birth defect syndromes, for this kind of approach. And so now this has now been done in a few babies, but it hasn't been done in a systematic way. So now, again, many of the sites around the world that were involved in the postnatal study, including Necker and some sites in California, are going to be doing this now in a prospective fashion. And so more to come on this, but I think again a very exciting treatment modality for structural birth defects in general. And so our lab had been working with these tabby mice, largely actually as part of thinking about the human study, and had access to some of the recombinant ectodysplasia, not the GMP drug, but just the one that you can make in the lab. And so since we had been working with these mice and thinking about the human phenotype, we also were thinking about what we could do in terms of using this in the mouse. And for this, we were very fortunate to collaborate with Jukka Jörnvall's lab in Helsinki. And Jukka is really an expert in dental morphology and particularly in terms of how we can understand the evolution of animals using the dental fossil record. And so this is a project that was led by Annie, a former student in Jukka's lab, and Kirsten, who was a postdoc in my lab. And we jokingly called this the Intelligent Design Project because the idea here was, could we intelligently design the shape of the tooth based on what we knew about signaling? And I'm just boiling down a large number of studies here into one image, in which the idea here was to see if we could take the extant wild-type mouse and revert its morphology to look like what we see in this rodent ancestor called Tribosphenemus minutus, which existed about 50 million years ago at the base of the rodents. And what we did, and this was first based on a number of computational analyses, and then it was taken in vitro, and then I'm showing you the culmination of that, which was the in vivo studies, was that we knew a fair amount about what the ectodysplasin mutation did in terms of cusp shape, and I showed you that on the previous slides. And we also knew that another signaling pathway called the hedgehog pathway, particularly a sonic hedgehog ligand, was important in affecting cusp shape and spacing. And so one of the main differences between the ancestor and the extant mouse is that in the extant mouse, these two cusps are connected by this crest, whereas that crest is missing in the ancestors. And so we then took a tabby mouse carrying an ectodysplasin mutant, and during mid-gestation, the same period when we were giving the recombinant ectodysplasin in the previous slides, Instead, now we gave an antagonist of sonic hedgehog. And indeed, what we were able to do was recapitulate this difference in terms of the separation of these two cusps as we see in the ancestor. And so this was a very nice proof of principle. And now, together with Yuka, we're thinking about how we can move this into looking at other signaling pathways and recapitulating other ancestral morphologies as well. And so for this first part, what we discussed was how XLHED represents the first structural birth defect syndrome with a potential medical treatment. And it brings to light these interesting parallels between human disease and evolution. And what I mentioned on the last slide here was how we're looking at these transitions in the fossil record. And of course, we can't prove that the signaling changes that we're making experimentally are the same ones that were important in the fossil record. But we think that by building up a series of different morphologies that we can recapitulate, that we can start to get a sense of what the signaling differences may have been during the evolutionary processes. And so for the next vignette, I'm going to switch into a syndrome, which is a rare syndrome that probably many of you have heard of, called Costello syndrome. And I'll just introduce to you the larger family of syndromes of which this is a part. And it was very nice to hear Guillaume's talk a little bit earlier, because I'm going to touch a little bit on this idea of modulating receptor tyrosine kinase signaling in vivo. So Costello syndrome is part of this large family of syndromes which we've seen an explosive growth in the discovery of over the past 15 or so years, called the RASopathies. And these are syndromes that are caused by activating mutations in the RAS-MAP kinase pathway. So this is a cartoon here of a typical RTK signaling pathway with a receptor binding the growth factor and then triggering signaling through various arms. And probably the best known of these is the RAS signaling cascade, which has two parts: the RAS-MAP kinase and then the PI3 kinase AKT arm. And what's been really interesting to see is how gain of function in many different parts of the pathway leads to syndromes. And you've probably heard of most of these. Probably the best known is neurofibromatosis. The two that we look at in our lab are cardiofaciocutaneous and Costello syndromes. And I'm going to tell you specifically about Costello syndrome, which is caused by gain of functions in HRAS, one of the family members. And one of the things that's really interesting to me about these syndromes, which I'm not going to talk about very much, but if there are questions we can discuss, is that these same mutations, in many cases actually the same exact mutations that you see as somatic mutations in cancer, when present in the germline, can cause these syndromes, but somehow these patients don't turn into one big tumor. They do have an increased risk of cancer, but it's actually, in some cases like Costello syndrome, not that much above the baseline. So this is, I think, a very interesting aspect of these conditions. And so, and I forgot to mention, this work was actually done by Alice Goodwin, who was a graduate student in my lab and is now a faculty member in craniofacial orthodontics at UCSF. And so what Alice did during her PhD work was to visit these family meetings, because these conditions, especially at the time that we began working on them, there were only a few dozen or hundreds of patients described with these conditions. And so they gathered at these meetings from, many of them flew in from abroad, and we were able to study them together with specialists from many other fields. And the first thing that Alice did was to characterize the craniofacial findings in this multisystem disorder, and she reported on some of the findings that you can see here in this picture, including relatively large heads, this bitemporal narrowing, thick lips with a large appearing mouth, and low set and posteriorly rotated ears. And they also have manifestations in many other systems. And so, as I mentioned, clinicians from these other fields also attend to these meetings and study these patients. And Alice is actually was a DDS PhD student, and so the teeth in these patients had not been assessed in detail, and so she looked at them, and what she found was that they had many indications of increased enamel wear, of defective enamel, including these striated lesions that you can see here better in this UV flash, and indications of increased wear. And the controls here were actually the siblings who attended the meetings. And then Alice formed relationships with these families, and so she asked if after they left the meetings, if they would mail her back the shed baby teeth. And so she was able to then do scanning electron microscopy on them. And what she saw was that compared to the controls, that in the affecteds, the outer layer of the enamel appeared to be missing, which is probably what explains the increased wear that we see. And so then Alice wanted to understand mechanistically what the signaling defects were and how they lead to the enamel problems. And so for that, we were fortunate to obtain a mouse model with mutation at the same place as where the humans have it. And as you're all probably aware, in mice, the incisors, the front teeth, grow continuously. And so that means that we can actually study amelogenesis, the process of enamel formation, in the adults because they're continuously laying down enamel throughout their lives. And so these are the continuously growing incisors. The molars, like our teeth, have roots. And what Alice found was that compared to the controls in the HRAS mutants, the molars were missing this outer enamel layer, and the incisor enamel layer was also defective. And so based on this, she decided to take these adult mice and then play with the signaling and see which arm of the pathway, this RAS-MAP kinase arm or the PI3 kinase AKT arm, were involved in amelogenesis. And so she obtained inhibitors of both arms of this pathway and then dosed the adult mice, which, as I mentioned, continue to lay down enamel throughout their lives. So these are the untreated animals. Compared to the controls, the HRAS mutants have this defective enamel layer, and they have a number of different abnormalities, including enamel pearls or sequestrations of the enamel. And so then Alice took these mice and dosed them with the two compounds that I showed you. And when she inhibited the PI3 kinase arm, that did not lead to improvement of the enamel. So we still have these enamel pearls forming and abnormalities visible with SEM. But when she inhibited the MEK arm, that led to an essentially complete resolution of the defects, so very dramatic improvement. So that indicates to us that in terms of amelogenesis, it's really the MEK arm of the pathway which is important in terms of Costello syndrome. And so what Alice found, to summarize this, was that these patients have this enamel defect characterized by the demineralized spots and striations, decreased thickness, and increased wear. And in the animal models, the activated RAS signaling causes defects that resemble the human condition. And when she inhibits specifically the MEK arm, that rescues the enamel defect. And similar to what we heard from the talk from Guillaume, there are trials now around the world in these RASopathies to give inhibitors of MEK signaling as well as of PI3 kinase signaling. In humans, this is challenging in terms of amelogenesis because it happens so early, but we think that even in the case of the teeth, if the condition is identified early enough, perhaps even prenatally, that manipulating the pathway could lead to improvements in terms of the enamel as well as many other systems. And so for the third vignette, I'm going to tell you about another part of the craniofacial complex, and this is one that we've more recently become interested in, which is the oral mucosa. And for this, I really owe acknowledgement to another former student in the lab, Kyle, who also was a DDS PhD student and became an oral pathologist and then a faculty member at UCSF. And recently actually has left for industry. But Kyle, when he was in the lab, kept coming to me and saying how interesting the oral mucosa was. And not being a dentist, I hadn't really thought about this very much, but as you all know, when you bite the inside of your mouth, it heals very rapidly, and unlike when you cut your skin, it doesn't scar. So it has very interesting healing properties. And because of this, it's used a lot in tissue engineering and other kinds of medical applications like transplants. One point that I need to make before I tell you about the work that we did is that we need to remember that the oral mucosa is very highly regionalized. And I'm going to focus specifically on the buccal or cheek mucosa. But if we look at other parts of the mouth, like the gingiva or the palate or the tongue, we see different kinds of behavior. So I'm going to talk specifically about the buccal mucosa. And this tissue is important in many rare oral diseases as well as in a number of cancers. And I put this image up, which was from work that Silvio Goodkin did when he was at the NIH, for you to see how these were in volunteer patients when he wounded the skin versus the oral mucosa, how much more rapidly and scarlessly the oral mucosa heals. And then this is from a review that Kyle wrote actually before he began his PhD. And just to show you histologically, the epithelium of the oral mucosa here has all the proliferation happening in the basal layer, and then the cells differentiate and then are sloughed off. And so the first work that we did was looking specifically at the epithelium, and then I'm going to tell you just for a slide or two about that because it's all work that we published, and then I'll spend the rest of the time telling you about our unpublished work looking at the role of the mesenchyme in wound healing. And so in terms of rare diseases, these largely fall into the category of autoimmune diseases, which cause these chronic and recurring ulcers. And these are very painful and debilitating, and they can lead to other health issues. So it's very important to treat them quickly and effectively. And many of these conditions shown here don't have currently very effective treatments, and it's actually quite challenging for the patients. So what Kyle wanted to do when he began his graduate work was to first understand the behavior of the oral epithelium. And he did a number of experiments. Again, this is work which has been published, so I'll just review for you what he found. This is an experiment that he did using this keratin 5 TTA histone 2B GFP label retaining system developed by Doina Tumbar when she was in Elaine Fuchs's lab, in which all of the epithelial cells are labeled in green. And that green label is continuously produced until we shut it off. And once we shut it off, it's then diluted in those cells that are rapidly proliferating and retained in those cells that are quiescent. And so what you see here is how rapidly the label is diluted out. Within about a week, it's disappearing, and within about 3 weeks, it's almost gone. And so what this shows us is that this tissue actually has one of the fastest turnover rates in the adult mouse, second only maybe to other tissues further down the GI tract that we'll talk about in a minute, like the esophagus and the gut. And Kyle spent a lot of time understanding the division kinetics of this tissue and really thinking a lot about the epithelium. But what we realized after working on this for a number of years was that if we wanted to understand the scarring properties or lack of scarring that we see in this tissue, we needed to think also about the stroma, the mesenchyme. And so that's work that is currently being done by Jesse, who's another graduate student in the lab, who's trying to understand this process of wound healing and relative lack of scarring in the mouth by thinking about the different parts of the process, including the initial production of extracellular matrix, which fills the wound, and then the signaling and tissue remodeling that occur after this. And I should mention, it's not that the oral mucosa doesn't scar at all. If you make a big enough wound, you will get a scar, but it's much, much less than what you see in the skin. And so the question that Jessie is asking in her graduate work is, how can she resolve the identities and functions of the fibroblasts in the buccal mucosa, and then think about the cellular and molecular differences between the skin and the mouth to understand why one of them, the oral mucosa, is so much better at healing than the skin. And this is a very close collaboration that Jesse's doing with Michelle Griffin, who's a postdoc in Mike Longacre's lab at Stanford. And what Jesse and Michelle have done is this series of matched 2 millimeter punch biopsies from the mouth and the facial skin at a series of time points from the initial wound afterwards for about a week. And the way that Jesse and Michelle have begun to characterize this is first by doing a single-cell RNA sequencing experiment in which they look at all of the fibroblasts. I should mention they're gathering all of the tissue in the mesenchyme, but right now they're focusing initially on just the fibroblasts, and then we'll look at other components like the vasculature later. And so here I'm showing you all of the cells from all of the different time points of the oral mucosa and the skin, but each of these cells is tagged with a barcode, and so we actually can go back and look for each of these where its origin was, both temporally and spatially. And so we then take these cells and overlay here first their origin, the oral mucosa being in red and the facial skin being in black. And what you see is that each of the 9 clusters has various composition in terms of its origin, which makes sense because they have different behaviors. And we see that when we look at the UMAP, that there's more cells from the oral mucosa on the right and more cells from the skin on the left. And this will be important in the coming few slides. So we see also, we can look not only at their geographic origin, but also at which time point after the wounding they come from. And that's shown here in this graph where we see unwounded postoperative day 1, 2, 4, and 7 from both the mouth and the skin. And what to me is interesting is how dynamic this is. So the proportion of cells in each cluster shifts very much over time, which tells you that there's a lot going on during this healing process. And I'm going to show you also on the next couple of slides how when we look at the UMAPs from the mouth and the skin, we can then begin to get some different trajectories in terms of the relationships between the cells. So here we're doing pseudo-time analysis, which predicts the future state of the cells, the trajectories that connect the cells over time, over this pseudo-time. And what we see is that, as I mentioned, on the left there are more skin cells, and on the right there are more oral mucosa cells. So we begin to see the emergence of a scarring versus regenerative trajectory on this UMAP. And we then can look into each of those trajectories and begin to pull out which genes and pathways are important in scarring versus healing. And we defined a few criteria in order to identify enriched pathways, including being upregulated in the oral mucosa versus the skin, being involved in the regenerative trajectory from the pseudotime analysis, and then using various network analyses programs like CellChat to identify pathways that are important. And based on this, we've identified 3 main categories. I'm just going to focus on the one that's most exciting to us right now, which is a pathway called the Gas6 AXL pathway. AXL is a receptor tyrosine kinase, and Gas6 is its ligand, and it's been primarily studied in the context of the immune system. Relatively less work has been done looking at epithelial-mesenchymal signaling. And so we see that the AXL receptor is upregulated in the oral mucosa compared to the skin at all of the different time points. We also see that both the Gas6 ligand and the AXL receptor are overrepresented in this healing or regenerative trajectory on the right compared to the scarring trajectory. And then when we use network analysis, what we see is that this Gas6 AXL signaling network is highly utilized for communication between the fibroblasts and other cell types, and more so in the oral mucosa than in the skin. So based on the initial computational analyses, there's a suggestion that this Gas6 AXL pathway might be important. But of course, this doesn't tell us if it is. We need to then go in and do functional experiments. And I'll say right now we're still in the process of looking at the mouse genetics, but we have some very promising preliminary data that I'll show you using small molecules. And so Jesse and Michelle looked using, this is a single molecule in situ hybridization, and we see that the Gas6 ligand is expressed in both the epithelium and the mesenchyme at much higher levels in the oral mucosa compared to the skin in the unwounded state. And then when we look at the percent of fibroblasts in the wound bed that are either expressing AXL or the activated form of AXL, we see that at all of the time points, unwounded as well as post-operative days, it's much higher in the mouth than in the skin. So again, looking promising, but the important test is what happens when we either inhibit or agonize this pathway. And so we began by injecting an inhibitor of AXL, a small molecule, into the unwounded state. And what we see is that in the buccal mucosa, compared to the control, where there's a very healthy, thick layer of epithelium and mesenchyme, both of these become atrophied after inhibition of AXL. So that indicates to us that this pathway is important in homeostasis. But what we really want to know is what happens after wounding. And so we were very encouraged to see that when we inhibit AXL in the mouth, which in the control by about 4 days after wounding has fully healed, that we see very poor recovery in the AXL-treated mouse. So this indicates to us that activation of this pathway is important for wound healing. And then, to me, an even more exciting result was when we looked in the skin at what happens when we inject the Gas6 ligand. So here, after 4 days, in the untreated animal, there's still a big wound. It has not yet fully healed, but we see much improved resolution of wound healing after giving the Gas6 ligand. So this suggests to us that this could actually be used therapeutically in patients. And so where we are right now is with this preliminary model that the reason, or an important reason, that we see much higher levels of scarring and fibrosis in the skin compared to the mouth is that there are higher levels of activation of Gas6 and AXL. And I didn't get into this in detail, but we think that one of the important roles of this higher level of activation is that these inhibit focal adhesion kinase, and that leads to lower levels of fibrosis. And there are hints that there actually may be a reciprocal interaction between focal adhesion kinase and AXL in both the face and the skin. And so this is something that we're pursuing further right now. And I'll also mention that recently a colleague of mine, Yache Su in Boston, published a paper showing that Gas6 is important in hair follicle stem cells. So it seems to be that there's this theme emerging now for a role of this pathway in this in epithelia and mesenchyme beyond its previously described role in the immune system. And so for the last few minutes, I'll tell you about the final vignette, which is how we think about the potential roles of understanding fundamental principles of intestinal stem cells in order to move towards therapy. And so, as I mentioned, the intestinal epithelium is a very regenerative tissue. It perhaps is one of the few tissues that turns over even more quickly than the oral epithelium every several days in both mouse and human. And in the small intestine, the business end are the villi, which is where absorption of nutrients and secretion of hormones and other substances happens. And then the crypts, shown here in cartoon form, are where the progenitors are present. And these cells turn over every several days, terminally differentiate, and then get sloughed off the tip of the villus. And about 15 years ago, a very important discovery was made by Hans Klever's lab in the Netherlands, identifying a gene called LGR5, which is a member of the Wnt signaling pathway, as a marker for a cell type called the crypt-based columnar cell, which are these thin, rapidly proliferative cells that are sandwiched in between secretory Paneth cells at the crypt base. And these LGR5 expressing cells have been established as the workhorse stem cells of the intestinal epithelium. And we got into this through a collaboration with Fred de Sauvage's lab at Genentech. And we worked with Fred's lab to study an allele that they made in which they had knocked in a diphtheria toxin receptor together with a GFP reporter to the LGR5 locus. And this is showing you in low magnification that all of the crypt-based columnar cells express this construct. And when we look at higher magnification, what we see is that these LGR5 expressing cells are proliferative, as indicated by expression of Ki67, as are their descendants, which are the transit amplifying cells. And so when we began this project, our hypothesis was that ablating these cells by giving the diphtheria toxin would have catastrophic effects for the gut, because we knew that these LGR5 expressing cells were so important for the turnover of the epithelium. And we were very surprised to find that actually the intestine looked totally fine. After, this is 24 hours, 10 days, so 2 rounds of renewal, histologically and every other way that we looked, the gut really looked perfectly fine. And yet, when we withdrew the toxin, the LGR5 expressing cells rapidly reappeared. So here we're giving the toxin for several days. There's no LGR5 expressing cells. Within a couple of days, they're coming back, and within a few days, they've repopulated the base of the crypt. So the gut likes to have these cells there, but it can very well tolerate their absence. And so this is a nice example of plasticity, which is a topic that we've been very interested in studying in the lab. And we want to learn, how is this regulated, this amazing plasticity that we see? How do the stem cells respond to injury and inflammation? And then in the long run, can we harness the power of this plastic behavior in order to treat diseases? And so the rare disease that I'm going to focus this last part on is inflammatory bowel disease. And I chose this because this also, as many of you know, has important oral manifestations as well. But I'm going to first tell you about the model that we're using, which is worm infections of the gut. And the reason that we got interested in this from a basic science perspective is that these worm infections infect these worms infect billions of people in the developing world and virtually all animals in the wild. And while they're generally not fatal, they can cause a lot of morbidity. And in many cases, though, they're actually not non-symptomatic. And these infections have been largely eradicated in the developed world, which is mostly a good thing, but it has some unintended consequences related to this hygiene hypothesis that we develop in an overly sterile environment, because of the idea that there has been, with this coevolution of the worm and the host, this affects the immune responses. And so the worm infections may actually protect against some immune-mediated diseases like inflammatory bowel disease. And you see reports of patients who will travel to places like Thailand and Malaysia and actually self-administer these worms in an attempt to shift their immune balance in order to treat their disease. And so what we wanted to do was understand, how are these worm infections affecting the gut, and can we think of a more mechanistic way to treat these diseases as opposed to just feeding patients worms? And you can see on this map, this is correlative, but there's an inverse correlation between where there's a high level of autoimmune disorders and where there's a high level of helminth infestations. And so we think a lot about conditions like early onset pediatric IBD, which is this rare disease with very severe complications and poor treatments. So the model that we use is this natural mouse worm infection called Heligmosomoides polygyrus, which as part of its life cycle, the larva will invade through the wall, this is the crypt here, and live inside the mesenchyme for about a week. And it's encircled by this granuloma consisting of myeloid cells. And this here in the H&E image shows you the worm snaking in and out of the plane of the board. And what was known when we began this project was that the crypts that are present above this granuloma are hyperproliferative. And so that suggested to us that something either made by the worm or by the immune cells was hyperactivating the stem cell program in these crypts right above the granuloma. And so this is a close collaboration with Rich Loxley's lab at UCSF. It was done by Ice Brown, a former student in our lab, and Adam, former postdoc in Rich's lab. And what Ice Brown and Adam did was to infect these LGR5 reporter mice, the same ones that I showed you on the previous slide, with the larvae. And they got this very surprising result, which was the opposite of what they had expected, which was that rather than seeing hyperactivation of the stem cell program, they actually saw that the LGR5 reporter was turned off in those crypts near the granuloma compared to those crypts at a distance. And it wasn't just LGR5. They then looked at a surrogate marker of the stem cells, which is a gene called OLFM4. This is by in situ hybridization. And again, we see here that expression of this gene is turned off in the crypts above the granuloma compared to those crypts at a distance. And so at this point, what Ice Brown and Adam did was to take an unbiased approach by punching out hundreds of these little granulomas, sorting out the crypt-based epithelium, and then doing a bulk RNA sequencing experiment. And they got this very nice data set, which showed us that not only so on the left here are the granuloma-associated crypts, and on the right are the crypts that are at a distance. And so it wasn't just LGR5 and OLFM4, the two genes that I showed you on the previous slide, but actually all of these genes in green here that are downregulated in the granuloma-associated crypts constitute the transcriptional signature of the intestinal stem cell. So it's like there's a wholesale shift away from the normal gene expression program towards something else. And indeed, we see that there are a lot of genes that are upregulated in the granuloma-associated crypts. And one that we focused on was this gene called Ly6A, or SCA1, which at the time had not really been explored very much in the context of the gut, but is known to be expressed in a lot of adult stem cells. And so there's a nice antibody for this, and we see that SCA1 is upregulated in those crypts near the granuloma, where LGR5 is turned off compared to the crypts at a distance. And so we can use this to sort out these two populations by flow cytometry and then put them in culture. If you take LGR5 expressing cells, the homeostatic intestinal stem cells, and put them in culture, then you get structures called organoids, which are like mini guts, which have crypts and express many of the cell types that you see in the adult gut. And indeed, that was what we saw when we took the SCA1 negative cells, the LGR5 positive cells. They formed these typical budding organoids. But when we put the SCA1 positive cells in culture, we got this result that we didn't know how to explain at first, which was that they formed these very large cystic spheroid structures. But then we looked in the literature, and what we saw was that if you take embryonic mouse cells from the embryonic mouse gut and put them in culture, then this is exactly what you get. And so that suggested to us that perhaps what's going on here is that the gene expression program is shifting away from the adult program towards something more like what you see in the embryo. And indeed, when we looked at gene expression in the spheroids compared to the organoids in culture, as well as looking back at the in vivo data set that I showed you before, we saw that they had both upregulated fetal gene expression programs after the infection. And so that led us to propose that what's going on in these infection models, and this has now been studied by several other groups and several other injury models beyond just infection, like irradiation or giving dextran sodium sulfate, which mimics the inflammation seen in some forms of IBD. They've also found similar results, which is that while in the wild type or unperturbed gut, we see that the LGR5 expressing cells are the ones that fuel the renewal. And in certain cases, like if we ablate the intestinal stem cells with mild forms of damage, we can get this dedifferentiation of committed progenitors that happens. That there's also what we call developmental reprogramming happening, where you shift the adult gene expression program towards something that looks more like what you see in the embryo. And that, for reasons that we don't yet understand, seems to improve the ability to recover from various forms of damage. And then after the healing happens, you then shift back towards the adult gene expression program. And so I'll just mention one other example of plasticity that we recently published, which is looking at the role of the blood and lymphatic endothelial cells in the gut. And this is work that was done by two postdocs in the lab, Jeremy and Brisa, who were interested in looking at how this lymphatic vasculature, which surrounds the crypts, may also have a role in this plasticity that we see during healing. And so here I'm showing you a couple of images demonstrating how close the lymphatic vasculature is to these LGR5 expressing cells that I mentioned before. And what Jeremy and Brisa did, and I won't show you all this for the sake of time, is a single-cell RNA sequencing experiment, and they found that there are very high levels of R-spondin-3, which is one of the ligands for the LGR family of proteins. And so we see that R-spondin-3 is expressed at very high levels in this lymphatic vasculature, but it's also expressed in several other cell types in the mesenchyme. And so they then knocked out this R-spondin-3 in the lymphatic vasculature using the PROX1-CRE, which is expressed in those cells. And initially we're disappointed because we didn't see any effects on proliferation or many of the other markers that we looked at. And so we then asked whether, because there are several sources of R-spondin, and this gets back to this theme of plasticity, that there may be redundancy in the crypt. And so what they then did was give 5-fluorouracil, a chemotherapeutic. And here what they see is that there aren't major effects on weight loss, but when we eliminate the R-spondin-3 in the lymphatic vasculature and give 5-fluorouracil, that there are now effects on proliferation as well as on, so this is quantified here, as well as on the number of stem cells as shown by OLFM4 and LGR5. And so I just wanted to show you this other recent example as a model of this plasticity that we see in the gut, in which there's this hierarchy of requirement for R-spondin-3, where during homeostasis, there's no effects on proliferation if we eliminate the lymphatic R-spondin-3. In mild forms of injury, like ablating the LGR5 cell, similarly, this is well tolerated. But once we get to higher levels of injury, like 5-fluorouracil, this chemotherapeutic, then there is a role for the other sources of R-spondin-3, like that in the lymphatics, and that's summarized here in this cartoon. And this is a theme that we're beginning to explore further. Is first of all, how is the vasculature important as a source of signals to the stem cells, and more generally, how are these different sources of growth factors that we see in the mesenchyme important for regulation of the intestinal epithelial stem cells? And so with that, I'll stop. I mentioned all of the folks who did the work as well as our collaborators. As I was going through the talk, I want to acknowledge funding both from the NIH as well as from foundations for this work. And I'll stop there, and I'm happy to take any questions.
Thank you very much. So it was a very rich presentation, so many questions may be raised. Please.
Thank you for a very nice presentation. I was especially interested in your research on wound healing. So you looked at oral mucosa and skin. And for example, you showed the results about the kinase, the FAK, and how that influenced the fibroblast growth. And do you have any insight on how that relates to hypertrophic scarring?
Yeah, it's a very good question. So that would sort of be the opposite of what we're looking at right now, and it's our collaborator Mike Longacre has a long history of studying wound healing, so it's definitely an area that we want to get into. Right now, we haven't looked at that yet, just because of time. But the question is whether inhibiting this pathway or perhaps some of the others that we are beginning to identify may enable us to prevent or even cure hypertrophic scars. And Mike's lab has a lot of work in this area, looking at various pathways like the hipposignaling pathway and others as well. So yeah, and then you can get into a lot of other questions like, is it important to inject it early or late, and things like that.
Thank you.
Hi, Ophir. We are also interested, as you know, about oral mucosa wound healing. And we didn't use the same model than you because we worked with human tissue on wounded. And we didn't identify the same pathways than you. So I'm wondering how you did choose your pathways of interest. How did you choose them? Did you take the most upregulated or just the one that you knew were of interest for wound healing?
No, yeah. So as I mentioned, we developed these because we get a lot of data out of this, and there's going to be work in there for many labs, to look at in the future. So we try to initially narrow it down computationally, and we ended up like basically looking for things like is it preferentially expressed in the mouth versus the skin, and we then ended up with 3 different categories. Or 3 different kind of main groups of pathways, and this axle one, there was nothing known about it, so we thought, it's worth it's worth looking at. But I don't want to imply that means that it's necessarily the most important one, and we won't know until we actually do the functional studies. And it may be that some of them work together, and that by modulating them in parallel, that we will get even better effects. So I think that it's going to open up a lot of really interesting questions. And I also think that comparing it to the human is going to be very important.
Yeah, I totally agree. And one last question. At the beginning of your talk about intestine stem cells, you showed us also some oral mucosa diseases like Behçet's. So did you have any effect on helminth infection or on oral epithelium?
I'm not an expert on this, but my understanding is that the patients who have undergone these worm treatments see improvement of all of the aspects of IBD. As far as I know, there haven't been a lot of studies yet on how this affects the mouth, and we haven't done that either. One little caveat to the study that I mentioned is that all the work that I showed you, comparing, for example, the RNA-seq of the LGR5 positive cells and the cells that have turned off LGR5 is from infected animals. So that's basically looking at short-range effects of the infection. There's also probably systemic effects. So in order to study that, we would need to compare infected and uninfected animals, and we haven't done that yet. But and there, you can take this. There's a lot of interesting things you can do. Like there's other models of worm infection that don't cause granulomas but still infect systemically. So you could compare those, and we just haven't done it yet. But it would be interesting to do that.
Going back to your first theme, which was the ectrodysplasia story, which is from Europe to States. And, it's like an economical pitfalls that you mentioned. Mm-hmm. Could you give some new or what will happen soon or in late stage?
So
is it continuing?
Yeah, so there's, really it's thanks to Holm Schneider doing this experiment in prenatally that there's this organization. It's not exactly a company. It's called Esperare, and my understanding is that they're sort of a nonprofit, but they're trying to develop this as a therapy, and they're working with a company to make more of the drug. And they have several sites, and there's an ongoing trial. There's sites both in Europe and the U.S. I think it's been a little bit slow, in large part. Identifying families is tricky because in order to be a candidate for this, you have to you have to know the mutation that's running in the family. So it has to be a family who's already had an affected baby, and then be identified early enough in the pregnancy that you can do the testing, see if the fetus is affected, and so it's not a trivial trial to do, but I'm very encouraged by the preliminary studies, and I think it's very exciting and can lead to a paradigm shift in how we think about treating these kinds of diseases.
Yes, and you mentioned that it was like a revolution in the concept, and other people said that from him to surgery to medicine, So there was a proposal, which I think is very provocative, is to test born babies, and if you have a therapy which could cure the developmental defects, it's less costly than the whole pathway that the patient has to have when you have a developmental defect. So I think that the way we think personalized medicine and inborn error in pediatric and during pregnancy will be like a revolution in some years.
Yeah, there's even a fair amount of activity now in the prenatal treatment space where people are doing fetal exomes and identifying conditions, and we can talk later. I mean, there's a lot of promise and a lot of very important caveats and ethical issues that arise from these kinds of approaches.
But our way of thinking will be like modified.
Yeah, I think so, yeah.
So I thank you very much. Do you have any question? Anne?
Yes, I have a small question. Thank you very much. It was really interesting. Really interesting about the disease linked to RAS pathway, because as a maxillofacial surgeon, we treat patients with giant cell granulomas of the jaws. And did you observe in the mouse model of Costello syndrome such lesions of the jaws or of the long bones?
We focused really on the teeth initially. Now we're looking at the skull in this model. We haven't looked at the long bones, but I think that there is a paper that looked at this. I can't remember exactly what they found. We've started looking at the skull base, and we see some very interesting differences that we think might explain some of the craniofacial shape differences. We're not quite done with it, but we're getting there.
Thank you.